Thank you so much to the organizers for the invitation to participate in the Biophysics Awareness Week. I'm really excited to tell you a little bit about my work, but also about my journey into science and biophysics. My name is Rogelio Hernandez-Lopez, and I am finishing my postdoc in the lab of Wendelin at UCSF. And I am about to start my research group in the departments of bioengineering and genetics at Stanford University. Let me ask you a very simple question. Are you thinking of pursuing a career in biophysics? So hopefully in the next few minutes, I can convince you that you should do it. And there is many very cool problems and techniques and approaches that you can use to understand biology. And I want to share with you some of the things that I find really fascinating about this field and the problems that I think are very cool to work on. One of the main reasons that I think decided to enter science, but later also enter this field of biophysics is that you can live and travel to different places and meet very talented people. So I was born in Mexico City, but raised in Oaxaca in the south of Mexico. And I think a very important moment for my decision to enter science was my participation in the International Chemistry Olympiad. I had the opportunity to travel for the first time outside of Mexico. I visited Greece, and I met people who are still my friends and professors and other people who I am still in touch with. That experience made me decide to move to Mexico City and enter college. I did my degree in chemistry. And while at the beginning of my college degree, I was doing research in chemistry, towards the end, I wanted to explore physics and math, but in particular, how you use those tools to study biological problems. So through conversations with friends and professors, I decided to enter the program of chemical physics, and in particular, the Leshner lab at Harvard University. And I'm going to tell you in a moment some of the problems that I work on while I was a grad student. But after finishing my PhD, I moved for a brief time to San Diego and later to San Francisco to start my postdoc and switch fields to study now T-cell engineering and synthetic biology in the lab of Wendelin. And as I mentioned earlier, I'm about to move to Palo Alto and start my own research group. So, but really what motivated me and has motivated my research is the type of questions and biological problems that you can work on, in particular, that these expand multiple scales and approaches and techniques. So during my PhD, I work at the molecular scale, and in particular, on a group of proteins called microtubule-based motors. I'm going to tell you in a moment what is fascinating about these motors, but later I transitioned to the cellular scale and trying to understand T cells, immune cells, and how this can be engineered to, for example, kill cancer cells. And I am hoping to work later in a range of problems, but in particular, how collections of cells, multi-cell systems come together. I am really interested in the problem of the tumor microenvironment and how immune cells and collections of cells can be engineered to deal with tumors. So let me begin by telling you a little bit about the work that I did during my PhD. So this is a image of two cells where the nuclei are shown in green. And there is two types of filaments that are labeled here. In yellow, we have the microtubule filaments, and in purple, we have the actin cytoskeleton. These proteins are very important because they are the structure of the cell. They are helpful for cell division, but also they are the roads, the streets of a group of proteins called molecular motors. So these motors move along these filaments, and they are important for a variety of biological processes, but one of the most regarded ones is the intracellular transport. And two families that I work on during my PhD are adenines and kinesins that move in the opposite directions along microtubules. So I don't have a lot of time to tell you all the details of the work, but I got fascinated by this motor dyning, that moves along microtubules. It uses the energy of ATP hydrolysis. And using a technique called cryo-electron microscopy and molecular dynamic simulations, we were able to describe for the first time how is it that this motor binds and releases from the microtubule. What interactions are responsible for this process of binding and release. And then in collaboration with grad students and postdocs in the lab of Sam Rick Peterson, and using a technique called total internal reflection microscopy, you can watch and measure properties of these motors, such as their velocity, their run length, the amount of ATP that they consume when they move along microtubules. And not only that, but using all this knowledge of the very detailed information of the interface between the foot and the track, you can engineer these motors to have novel behaviors. For example, at that time, we realized that mutating some negatively charged residues for either a neutral charged residue or a positively charged residue will lead to dyning motors that now are moving 3 or 5 times more than the wild type motor. So something that I just want to mention is that, like, by using this biological problem as an excuse and using these multiple techniques, as I mentioned earlier, cryo-EM, TERF, and protein biochemistry, then you can learn about biology and also engineer new biology or engineer proteins with novel behaviors. Another problem that I've been working on more recently is in the field of T-cell engineering. And hopefully by now you have heard a lot about immune cells, but I was fascinated by this ability of the T cells to be able to search along our bodies, surveilling against virus, bacteria, and being able to launch very powerful killing programs to defend us. So due to the work of multiple groups and different techniques and approaches, scientists have realized that now these T cells can be engineered to kill cancer cells. And in particular, that they have the ability to, for example, infiltrate and kill tumors. And the idea here is that using synthetic biology and genome engineering, you can put new receptors in the surface of these T cells. And with these receptors, recognize particular proteins that are expressed on the surface of cancer cells. And in this way, you can redirect the cytotoxic activity of T cells against cancer. So this approach has worked really, really well for some diseases such as lymphoma and leukemia. And the type of questions or problems that I got interested have to do when trying to use the same approach, but now to treat a solid tumor or cancer such as breast or ovarian. Because the problem there is that there is not many proteins that are unique and that can be used to differentiate only tumor cells from normal tissue. As for example, the protein HER2 is expressed in your lungs and your heart, but it's expressed at low levels. And that protein is also present in tumors, but at high levels of expression. If you try to engineer a T cell against HER2, for example, there is actually problems of toxicity because these just normal CAR T cells, they don't have the ability to recognize or discriminate their targets based on the amount of the protein that is expressed on the surface of cancer cells or target cells. So what the goal of my research in the past few years was to engineer a sigmoidal response. In the field of systems biology, this sigmoidal response is called ultrasensitive response. And what we wanted to do was to engineer a T cell with the ability to only kill target cells that express high levels of a protein of your choice, but remain safe if you put these T cells in front of cells that express low levels of an antigen that represent or are characteristic of normal tissues. So we took inspiration from biological systems that have this ultrasensitive response, and I don't have a lot of time to give you all the details, but hopefully I can convince you by showing you these movies that, while the current CAR T cells do not have the ability to discriminate, as I mentioned earlier, they will kill both cancer cells or normal tissue, regardless of the amount of protein that is expressed on the surface of these cells. Now, by using a very simple design and a synthetic circuit to engineer these T cells, we can build smart T cells, cells that are able to only kill cancer cells, but remain safe against normal tissue. So you can read more about this work and other experiments in recent publications here, but I just wanted to mention that the strategy we used was using another synthetic receptor called synthetic notch receptor that controls the expression, the amount of the chimeric antigen receptor, the CAR molecule that is going to activate the T cells against their target. So by using these two receptors and playing with the affinity of these recognition domains against a protein of your choice, we were able to transition or to engineer this ultrasensitive response into T cells. So finally, I just want to mention for my future work, I would like to combine multiple techniques and approaches and really work across the scales from proteins, cells, and tissues. In the particular case, for example, of the tumor microenvironment, trying to understand how the structure of the tumor, the composition of it, and the spatial organization of the different components of these tumor structures affect the activity of endogenous immune cells or engineering immune cells, and how you can design and engineer novel approaches to make these therapies more successful. So please reach out if you are interested in the problems that I mentioned earlier, or you just are curious about biophysics, or how you decide on a question or problem to work on. This is my email address and my lab's website and my Twitter handle. My lab is gonna start at the Stanford BioX in the Clark Center. And with that, I would like just to acknowledge past and current advisors from college, PhD, and postdoc, and all the people that I have met and have interacted with the past few years, and the funding sources for the fellowship at support of my career. Thank you.